different enzymes, and hopefully with this kind of bioprocess technology, next year, I think next year we have to be able to scale up the enzyme production. Yeah. So please, Dr. Prabu, thank you for sharing your knowledge and also experience in this field. And as you may also know that this is our commitment to develop human resources in this bioprocess technology from this year and also next year. Yeah. So we have recruited some new students in bioprocess technology, and hopefully also our researcher can also improve their capacity by collaborating with colleague or partner from overseas. Yeah. So in here we have Dr. Prabu from India. So please also you may later on introduce yourself to our participants this morning. Yeah, okay, everyone. So please enjoy the webinar today, and hopefully we can gain more or insight, into the bioprocess technology, specifically for enzyme production. So I think that's all from me. Thank you, Pak Catur and Mbak Helen, for hosting this webinar, and so Mbak Lia and team, for arranging this webinar. Thank you, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Ahmad Fatoni, for the opening remarks. Before we move to the main session, we will have a photo session led by the committee. So we kindly ask all of the participants to turn on their cameras. And I will start with the first slide. 1, 2, 3. And the second slide. 1, 2, 3. Okay, now we will move to the main event today, which is presentation and discussion led by Dr. Catur Sri Herwanto. Dr. Catur, the time is yours.
Yes, thank you, Miss Helen. So, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. It is my pleasure to introduce our distinguished speaker, Dr. Masita Mani Raja Prabu, will be sharing with us a presentation strategies to enhance urease enzyme production from microbes for biocementation application. Dr. Prabu is an expert in biotechnology and microbiology. Currently, he is working as assistant professor in Department of Microbiology, Excel College for Commerce and Science. He has completed his PhD in microbiology at Department of Microbiology, Periyar University, Salem in India, and he has completed 2 master's degree. The first one is Master of Philosophy. Tes.
Maybe unmute, Pak.
Oh,
Dr. Prabu, you're still unmute.
okay.
Open your mic, maybe.
Okay, tes. Shall I repeat again? Tes. Can you hear me now?
Yeah, yes, sir.
Okay, shall I repeat again, right? Good. Yes, okay. It is my pleasure to introduce our distinguished speaker, Dr. Masita Mani Raja Prabu, will be sharing his expertise on strategies to enhance urease enzyme production from microbes for biocementation applications. So Dr. Prabu is an expert in biotechnology and microbiology. He is currently working as assistant professor in Department of Microbiology, Excel College for Commerce and Science. He's completed PhD at Department of Microbiology, Perrier University, Salem. So, Dr. Prabhu, without further ado, we would like to invite you to do your presentations. So the time is yours.
Yeah. Good afternoon to all. I'm Rajapur from India. Now I'm joining as a postdoctoral researcher in Research Center for Applied Microbiology in the Enzyme Production Technology Group. Herewith I share my topic. Let me talk about here is today's discussion on the topic of strategies for enhanced microbial urease production for biosanitation application. Myself is Dr. Raj Prabhu, and my host researcher and supervisor is Dr. Dadang. And the biosanitation is the process where the process of binding a soil particles through microbial activity, especially by the enzymes. And then now we focusing on the urease enzyme, which will be used to biosanitation application. Many microbes producing urease enzyme, and the urease will act and hydrolyze the urea substrate and forming carbonate ions, and which will be these carbonate ions will be react with calcium and to form calcium carbonate, which will be binding the soils together and strengthening the soils. These are these two methods by the biosanitation by microbes. So once the left one is the microbes used for the application of biosanitation, also the purified enzymes that will be used for the sanitation purpose. Why we choose move to the alternative biosanitation methods? Because the traditional sanitation methods will affect our environment by in the production time, and also the after the production it will be more affect the environment by polluting the environment. Also it reduces sanitation, and also global carbon dioxide emission, which will be polluted our environmental air, and also cause some illness to the surrounding surround system, and also in the both human and animal systems, and also causing air pollution and water pollution from the alkaline runoff from the sanitation industries, and also which leads to the resource production. Cementation product, cement production needs more limestones, and here it is why we choosing the biocementation is as a alternative methods to traditional cementation methods. Just in this biocementation production, it contains low carbon dioxide emissions compared to the cement production, and also it requires low energy to produce the biocement. And also we can use renewable biological materials to produce cementation products, and also it has some self-healing capability to heal the crack and then other repairs in the environment. Also it reduces the soil excavation and waste. Here it is many type of biosanitation methods are by the microbes. First one is enzyme induced carbonate precipitation. Here the researchers used enzyme from bacteria and our plant. It is enzyme induced plant, but we focusing on the microbe. And another is microbially induced calciate precipitation. Here it is whole microbial cells like ureolytic bacteria like Sporosalcina pasteurii used for the biosanitation. And then the third method is denitrification induced carbonate precipitation. In this method, researchers uses denitrifying bacteria like Pseudomonas and Paracoccus species to precipitate the carbonate precipitation to form the biosanitation process. And also the fourth one method is sulfate reducing bacteria induced biosanitation method. Here the use of sulfate reducing bacteria like D-sulfur species used for the precipitation to form the biocement. And then here is the mechanism of biocements by microbial urease. And then first reaction says in the presence of urea and water, the urease enzyme acts on the urea and releases the ammonium and carbonate molecules. When the carbonate molecules released, it will be binds and then react with the calcium. It will supply a supplement to the solution, and it will form calcium carbonate precipitation. And when we the calcium carbonate in the presence of soil grains will form the bridges between the soil particles and also binding the soils by using crystal formation. Here it is, we focus on the bacterial enzyme-induced carbonate precipitation. In this precipitation, we can use the crude enzyme extract from bacteria. Otherwise, we can use the purified urease enzyme to the precipitation method. Here it is the whole method. First, we have to prepare the urease enzyme and purification, and then you have to prepare the cementation solution with urea substrate and also apply to the soil or the other way when we recure the desired environment. And then urease hydrolysis process is carry on, and then when the urea hydrolysis process are done, then the calcium carbonate we follow fermentation process is strengthening and binding up enzyme-induced carbonation like bacteria in the high salinity and also high temperature. And compared to the bacteria, it will survive more than optimum temperature microbes. And then also enzyme work in the low oxygen surface. And also the fast reaction will be occurred when we using the purified enzyme faster than compared to the when we using the microbial cells in the MICP due to direct enzyme availability of will be leads to the faster reaction rate in this environment. And also we can control the chemical reaction by adjusting the chemical and enzyme concentration precisely. And also compared to the whole microbial cells, it will be very low cost to using the crude enzyme. It is inexpensive to produce, and also low carbon dioxide footprint to compared to the traditional cement. And then limitation of EICP, and then ammonium accumulation is a major button issues that will be after the urea hydrolysis, the ammonia will be toxic and also the created pollution in the environment. So the toxic and restrictive during the urea hydrolysis. And also we need another method to recure ammonia removal treatment. And also the enzyme, sometimes the enzyme may be affected by the environmental factors like pH, temperature, and other factors, cofactors. So the enzyme has short enzyme lifespan, so due to the environmental factors. And also limited penetration in low permeability soil like clay or sills type of soils. And also occurred the high calcium concentration, which will be enzyme inhibit the enzyme activity. When we use the high calcium concentration in the cementation solution, it will be affect the enzyme activity. Here it is strategies to improve the bacterial urease protection. Here we know that many methods like genetic engineering and then metabolic engineering and also basic microbial methods, genetic modification and then engineering of nickel metabolism. It will be reduced. It will be enhanced and improve the urease enzyme production. And also we can optimize the growth condition of bacteria, which leads to the more enzyme production. And also the immobilization of enzymes will be used for more directly used in the environment to the apply the environment to the biosanitation solutions. And we have to start, before we start, we have to select the ideal strain which exceeds the high use activity compared to the compared with many urolytic bacteria, and also it should be fast DS growth microorganism and non-pathogenic, because this pathogenic when we apply the pathogenic bacteria to the biosynthesis solution, it will be causing some disease. So we get this potential strain gas potential should be non-pathogenic, and then it also environmental tolerance to the desired environment. Also, we can produce more enzyme by optimizing the culture media by adjusting urea concentration, the urea concentration between the 0.25 to 1 molar, and then selecting appropriate calcium ions to using this using as a substrate to the substrate or a co-additional factor in the simulation solution. And also we can use the waste derivative calcium sources to improve the efficiency and reduce the cost. And also we can use the use of cost efficient substrate as a major source, major energy source for the bacterial cells to improve the enhance the enzyme production. It will be reduce the cost, like we can use agro wastes and industrial wastes as a substrate, main substrate to as a supplement for the supplement, and also nutrient-rich to the enhance the microbial production. When we use the agro-industrial waste, it will be supports the microbial growth, and also we can get more enzymes. Another method is genetic engineering approaches, and will be overexpression using of urease enzyme genes responsible for urease production, urease A, B, C genes. We can use the collect the urease. We can get the desired genes, and then we can modify the genes with the desirable disable organisms, and we can produce more enzymes. And also we can use nickel transportation in the environment by nickel transport in the urease enzyme production. It will be enhance the biofilm genes, and also it supports the biosanitation solution. And we can so you can induce the stress resistance pathway in the urease activity. It will be enhance the urease activity and also survival of the enzyme in the environment. And we can use the another method, immobilization of urease enzyme. It will be more suitable to the environment. When we use the immobilized enzyme, it will be more viable, and the durability of enzyme will be expanded in the environment. We can use the immobilization agents like alginate and polyvinyl alcohol and biochar, improves the survival and reduce the loss out of enzyme from the environment. And also it enhance the calcium carbonate precipitation in the desired location. And then we can use the additives and cofactors for enzyme activation. And nickel ions, it will be supports the enzyme function of urease. It enhances the urease activity to cleave the urea and hydrolyze the urea and forming the carbonate molecules. And also if you use magnesium and Fe ions, it will be enhance the urease enzyme activity. The magnesium ions to shapes the crystal morphology of calcium carbonate precipitation, and also the Fe ions enhance the biofilm activity by using the bacteria. And when we use the biopolymers as additional substrate or cofactors in these enzyme, it improves the binding of soil particles with one to another and also enhance the calcium carbonate binding the soil molecules. And we can, and another factors is to improve the enhanced urease production is the using of precipitation modifiers like amino acids surfactants and humic substance will be regulate the calcium carbonate crystal uniformity and nucleation. And here it is factors that controlling the enzyme-induced calcium precipitase method process. First one, the urease activity. When we use the higher microbial, higher amount of abundance microbial cells, it will be higher activity by the enzyme. And also the when we higher activities, higher microbial urease activity is occurred, the fast precipitation will be occurred, and then that controlled by the potential bacterial species. And also temperature, pH, and ionic strength of this also these factors also play a role in vital role in the EACP. In the factors pH, the optimal pH which should be between the 8 to 9.5. And then when we introduce the enzyme in the low pH, it will inhibit the urea catalase, and also the too higher pH causes poor calcium carbonate crystal quality. And we using the optimum temperature for urease enzyme activity of urease enzyme activity is 25 to 40 degrees Celsius. Beyond that, we have to alternate some. We have to evaluate the stability of enzyme. But the low temperature will be slow. When we apply the urease enzyme in the low temperature, it will be slower the reaction and also affect the whole process. When we apply to the beyond the more temperature, 50 degrees Celsius, it causes enzyme maybe cause enzyme denaturation. It will be affect the whole process. And then we also consider the concentration of urea and calcium ion concentration. The balanced concentration should be needed to the supplements as a optimum concentration. The higher concentration of calcium chloride will be maybe inhibit the enzyme and also reduce the enzyme reaction. When we supply the high amount of urease, which creates the excessive ammonia release, so it will be reached to the more toxic and then create some other environment. We can use the additives like improvers, like skim milk powder, which will be which stabilize the urease enzyme to longer compared to the without stabilizers. And then also we can use exopolysaccharides and proteins, which will be improves the enzyme activity and also the whole process of EACP. And then biopolymers, we can use the biopolymers. It will be enhance the improves the binding of side particles together. And then we have to check the permeability of soil to the flow to allow the flow of fluids in the environment. So we have to choose sand or poorly sand like that soils to use the as a substrate for the vegetation. And the whole process is poorly occurred in the clay and slate like soils. And strategies to improve the bacterial enzyme-induced calcium precipitation efficiency. So we can stabilize. First one is stabilization of urease enzyme. We can use skim milk and casein or soy protein as a stabilizer to stabilize the urease enzyme, and also and another one is another factor to improve the efficiency. Immobilization of urease, immobilized urease. We can, if we compare to the direct application of enzyme, the immobilized urease enzyme will be more adaptable and then releasing of on the target site of cementation work. We can use urease and immobilize the urease by using silica alginate or alginate or biochar. And then optimize, we have to optimize the urea and calcium ratio in the cementation solution, and we have to maintain the temperature, environmental and surrounding temperature between the 30 to 37 Celsius. It will be optimum temperature to urease activity. And also we have to don't do the direct one step injection. We have to use stepwise injection to to allow the precipitation uniformity, and we can alter or control the injection by using well-designed injection system to use to control the injection of urease and calcium ion solutions, which will be improves the enzyme distribution in the using flow enhancement. And also the hybrid biosynthesis, some researchers suggest that hybrid biosynthesis by using urease enzyme and also exopolysaccharide producing by bacteria, it will be combined together. It will be enhance the biosynthesis. In the application of biosynthesis, the biosynthesis will be applied to the soil stabilization and also to prepare the concrete cracks and the buildings, and also control the soil erosion in the environment where occurred more oftenly erosion of soil. And then we can use the urease biosynthesis application in the production of construction materials, which will be strengthening the construction material. And then challenges and limitation of this bacterial enzyme induced carbon calcium precipitation. The uneven enzyme may be distribution, so it will be another challenges, and the high cost of urea and then calcium sources, it will be high cost, and ammonia releases and environmental concerns. Some countries have limitations into releasing the ammonia in the environment, and also optimization required for large scale field use. And this is future directions. We in this future we will focus on the ammonia-free ACP variants, and also immobilized enzyme carriers for longer life to enhance the durability of fermentation process, and also hybrid biogeopolymeric fermentation methods, and application in low permeability soils, and then field scale injection optimization on the fermentation. Thank you.
Okay, thank you very much for giving us inspiring presentations. Now we invite all the audience to have questions or comments. So please feel free to do so by asking directly to Dr. Prabu, or you may also write inside the comment box. So please. Is there any questions? Okay, before taking any questions from the audience, maybe I would like to ask Dr. Prabu a question. So, could you please share information to us? Maybe there is a country or company who really already apply this technology in large scale. Is there any?
Yeah, the application in the large scale is still processing in the many countries like Japan and Australia and Singapore and China. And they using this enzyme-induced calcium precipitation to the liquefaction control to stabilize the soil to construction where the site of construction, and also in Australia, they're using the biosimilar plaques to the in the coastal areas. In China and Singapore, they're using also stabilize the in the trials biosimilar or bicrowded foundation in the coastal regions. By using this application of enzyme-induced calcium carbonate precipitation as a biosimilar material.
Okay, thank you. Before taking another question for the audience, would you please stop sharing, Dr. Prabu? for a moment, please?
Yes. Yeah.
So here we have Dr. Edi Wahyono who raised his hand. So please, Dr. Edi Wahyono.
Okay, thank you, Pak Catur. Thank you for your nice presentation, Dr. Prabu. So, in the biocement application, you will use urea, yeah. Okay,
Yes.
urea.
Yes.
So I just think about for the future, yeah. So if you successfully apply this technology, so you would create a competition between urea for agricultural application and biocement application. Yeah, like palm oil for biodiesel and for food. Yeah. And in your slide, you said that one of the limitation is urea is also high price, yeah, high price. So of course, if we have urea for agricultural application and also for biocement, I think it would be hard to decrease the price of urea. So what do you think about this competition actually in the future if you would like to use urea in large scale for the application biocement? And the second question is any information about if we are talking about the strength, yeah, the strength point of view between biocement and conventional cement, which one the stronger, and also which one have the long lifetime? Yeah, that's my question. Thank you.
Yeah.
Thank you, Edi, for the
Yeah.
question. So, Dr. Prabu, please.
Yeah, we can use compared to the commercially available urea substrate. We can use the alternate sources like that. Some industries releasing the ureas in that effluent. We can use the instead of commercial urea, we can use the biofree amount of urea. We can use that substrate of the urea enzyme production. We have to evaluate in this strengthening of biocementation cemented products compared to the traditional methods. So still the process is going on.
Okay. Clear, Dr. Edi.
Okay.
I see.
Okay, good. Yes.
The signal is not stable, so it's still unclear. But okay, we can talk later in our team.
Yes. Okay, the internet connection is maybe a little bit unstable, so hopefully we will have another discussion in another session with
Okay.
Pak Edi
Thanks,
and
thanks, Dr. Prabu. Thanks, Pak Catur.
Yes. Okay.
Thank you.
Okay. Next question, please. Is there any more questions? Okay, if not, maybe my question will be the last. So, Dr. Prabu, is your work now in Cibinong also related to this biocementation? If so, how is everything going? So,
Yeah, it's okay. Here is no problem. we started in the optimization of culture growth. Sir, do you hear me?
Yes, please go on.
Yeah, now we focusing on the optimization condition of urease producing bacteria and also urease enzyme production condition, like optimal condition of in the lab scale. So we are processing in this process, and also we have to now be checking the urease production by bacteria in the different temperature and pH ranges.
Okay, thank you. Is there any question from the audience? Good. If not, then we would like to thank you very much to Dr. Prabu for his inspiring presentations. And I hope all of us have gained insights about the applications of the urease enzyme for biofermentation. So now I give back to MC, Miss Helen, please.
Okay, thank you, Dr. Catur, and also thank you for the highly impactful discussion today, Dr. Prabu. Before we conclude, we would like to express our appreciation to you. So we would like to present certificate of appreciation. Maybe the committee will share the certificate. We are honored that you able to share your knowledge today. Hopefully this webinar session will be highly valuable for our participant today. So this is the certificate of participation. Thank you, Dr. Prabu. And also to conclude today, webinar, we would like to, on behalf of the committee, we would like to thank all of the participants today. Thank you so much, and see you on the next webinar session.
Thank you, Dr. Prabu, Pak
Okay.
Catur, Mbak Helen.
Okay. Thank you.
Thank you. Thank you all.